Chemical reaction of analyte produces product that can be detected by using optical methods. Changes in light absorbed, scattered, or emitted by these products are used to determine the concentration of the analyte. Change in concentration of ions are sensed as potential difference between two electrodes. Calibrator solution of known concentration are used to establish the relationship between the magnitude of an optical or electrical signal and the corresponding concentration of analyte. Quantitation of routine chemistry analysis is typically based on one of two measurement principles: measurement of light, photometry or spectrophotometry, or measurement of electrochemical potential, potentiometry. There are many variations of photometry and potentiometry, but all have in common that they signal the amount of light. Electrical voltage is predictable related to the amount of analyte in solution. Photometry. Photometry relies on measurement of light by photodetector. The light may be absorbed by a substance dissolved in solution. Absorbance. The light may be scattered or refracted by particles suspended in a solution. Turbidimetry or nephelometry. The light may be emitted from substances that absorb light at one wavelength and emit light at another wavelength. Fluorescence. Specific wavelengths of light are chosen for each analysis based on the properties of the substance being measured. A typical light source lamp generates a broad range of wavelengths of light. A visible lamp produces light of wavelengths from 400 nanometer violet light to 700 nanometer red light. An ultraviolet lamp produces light of wavelengths from 200 to 400 nanometer. To select the desired wavelength from the spectrum of light produced by the light source, a device called monochromator or filter are used. A monochromator disperses the light much like a prism disperses light and allows selection of a narrow band of wavelength to be directed through the sample cuvette. Cuvette self-made of optical transparent material that contains solution for analysis by optical methods. Here the wavelengths of the light, cosmic ray and the gamma ray, X-ray, ultraviolet, visible light from 400 to 700 is IR, the microwave, the TV, the radio, the electrical power. This is lower energy, higher wavelength, and here high energy, low wavelength. Absorbance: when an analyte has an intrinsic color or generates a color upon chemical reaction, visible light is absorbed when it passes through a solution containing the analyte or reaction products. The selective absorption of certain wavelengths of light from the spectrum of white light gives the solution its color. For example, a solution containing hemoglobin appears red because light in the green range of spectrum wavelengths from 500 to 600 nanometer is selectively absorbed, removed from the white spectrum. Measuring the decrease in green light that occur when passing through the solution give an indication of the amount of hemoglobin present. Here, show the configuration in this figure. The measure light absorbed, the difference between emitted light from the source and the light that reach the photodetector. Here the cuvette, Here's the light source. Here's the photo detector. Absorption, photometry. Simply source of light, pass through the solution, photo detector. Compounds that have no visible color often absorb light in the ultraviolet region, and this absorbance can be used in same way as absorption of visible light. The specific wavelength of light chosen is based on absorption properties of the compound being measured. As the amount of substance in solution increases, the relative amount of light that passes through the solution and reaches the detector decreases. The decrease in light is termed absorbance. A formula called Beer law describes the relationship between concentration and absorbed light for a given method A, E, LC multiplied by L by C, where E is the extinction coefficient, L is the length of the cuvette, C is the concentration, Beer law. And this is the relationship between the analytic concentration and the change in absorption, the linear one. Turbidometry and nephelometry. Some tests are based on formulation of insoluble particles that interfere with the passage of light through the solution. The analyte reacts with an added reagent to produce insoluble particles that remain suspended in the solution. When light hits these particles, some of it's reflected in the different directions. A higher concentration of analyte presents a greater number of particles that will inhibit light passing through the solution and increase the amount of light reflected. It's possible to measure the loss of light passing straight through the solution called turbidometry, or the increase of light reflected in the different direction called nephelometry. In turbidometry, the detector is placed in a direct line with the incident light, and the light sensed by the detector decreases as the number of analyte particles increases. In nephelometry, the detector is placed at an angle to the light path to avoid detection of light passing through the sample. The nephelometric detector senses light scattered by the particle. The amount of light reaching the detector increases as the number of analyte particles increases. Often antibodies are used with these methods and represent a type of immunometric assay, specifically immunoturbidometry and immunonephelometry. The antibodies in the reagent will cause analyte molecules to form complex or lattices, and these large particle aggregates enhance the reflection of light, increasing the analytical signal that is measured. Here, turbidometry, cuvette containing suspended particles, the light source, photo detector. Here the light source, nephelometry, cuvette containing suspended particle, and the photo detector. Turbidometric and nephelometric methods are often chosen to measure proteins such as transferrin, Turbidometric and nephelometric methods are often chosen to measure proteins such as transferrin or biliary albumin. Biliary albumin, two important transport proteins in the blood. Proteins are relatively large molecules that can be easily cross-linked by selective antibodies to produce aggregate particles that are the right size to reflect light in the visible or ultraviolet range. Some drugs of abuse tests can quantitate the concentration of drug in the patient sample by measuring the change in turbidity due to the competition between drug in the patient sample and drug bound to microparticle with antibody added to the solution. Fluorescence. Certain kinds of chemical structures are able to absorb light of one wavelength and emit light to another wavelength. These substances are termed fluorescence compound or fluorophore. In each case, the incident light is of shorter wavelength and higher energy than the emitted light. So, a substance that absorbs blue light wavelength 400 may emit lower energy green light wavelength 500. The detector is placed at 90 angle from the incident light so that it detects only emitted light and not residual incident light that pass directly through the sample or reflect light bounded back from the sample or covert. Here, the incidental light is a shorter wavelength than the emitted light. Shorter wavelength, and this is longer wavelength. Analytes of interest in clinical chemistry are not entirely fluorescent. Instead, fluorescent molecules are incorporated as reagents to help detect analytes. For example, immunological methods for measuring the tumor marker like CA125 ovarian cancer marker and CA15-3 breast cancer marker use antibodies that have a fluorescent compound attached. The antibody recognized and bind to the tumor marker. Excess unbound antibodies are washed away and the amount of fluorescent light generated is in direct proportion to the amount of tumor marker in the sample. Chemiluminescence is light emission due to chemical reaction similar to fluorescence. There are some molecules that due to their structure can produce light rather than heat when they react with other molecules. Some immunological methods for measuring hormones and the tumor marker utilize chemiluminescent molecules. After a selective antibody bind to analyte in the patient system, the bound and unbound antibody are separated and by washing, and second antibody linked to the chemiluminescence molecule is added. The chemiluminescent molecule is added. Once bound to the first antibody bound complex, a chemical is added to the mixture to generate chemiluminescent signal that is proportional to the amount of analyte in the sample. It's used in ELISA technique. In a variation of technique and others, in a variation of this technique, the chemiluminescent signal is generated by pulsing the mixture with electrical current rather than through chemical reaction. This method is called electrochemiluminescence.